Our next speaker is Pam Ruegg from Michigan State. She's going to get 45 minutes. Luckily, we have a long lunch.
Thank you. I always appreciate that there's so much interest in subjects. And of course, previous speaker had the technical difficulties. So give you a break on that. So today I'm going to talk about a subject that is both near and dear to my heart and is also controversial, and which is kind of fun to do. And so I'm going to be speaking about where we're at today, really looking at the bovine milk microbiome, and present some of the conflicting and contrasting views on this subject. I want to start off by it's fun to be here. You know, I've got this little slide up here from one of my collaborators, Alessia, from Italy. And the thing that I have enjoyed is listening to people with enthusiasm about how cool it is how milk is produced. I think it's unbelievably cool how milk is produced. You know, it's a highly specialized process. It's not easily replicated. We don't even know entirely what's in milk. I think that is super cool. And anytime any one of my grad students comes in and says, I want to do this new technique, you know, that's been done in other substances, and I'm going to do it on milk, I'm like, okay, add 6 months to your graduate studies, because you're going to be stuck in the laboratory with unforeseen problems forever. And that always comes out like that. So producing milk is outrageously an important thing. It's a highly important source of high protein and nutrients for human nutrition. And one of the most important things that we need to do is maintain the mammary gland health. And maintaining mammary gland health is based upon prevention of mastitis, because it's a really interesting disease that affects actually everything about the process of producing milk. Now, mastitis itself is a widely studied disease, but it's widely misunderstood. And I say that as somebody who's spent my career working in this area. Mastitis is primarily a bacterial disease of the mammary gland, but its detection is based on observation of inflammation in the milk and or the udder. And it's got bacterial etiologies, but we don't always find the bacteria there. And so a lot of people then assume that mastitis is a single entity as a disease based on the detection of that inflammation. But mastitis is caused by a wide variety of bacterial etiologies which have unique pathogenesis to them. And so that's where the difficulty comes, because it's often detected based on inflammation. The inflammation is a nonspecific response to infection by a wide variety of bacterial pathogens that require specific intervention depending on what those pathogens are. So it's a pretty interesting disease, widely misunderstood. Infection of each mammary gland is independent, although there is inflammation which occurs in all the mammary glands regardless of infection. And the process of actually becoming infected requires these bacterial infections to overwhelm both anatomic and immunological defenses. And the most interesting thing of all is probably the most common outcome of any bacterial infection of any mammary gland is spontaneous bacteriological clearance. And we don't recognize that at all. But if you've ever looked at individual somatic cell count or any sort of immunological biomarkers of inflammatory response at the mammary gland, there's a lot of activity there. Mammary glands getting infected all of the time in an immunologically healthy cow. They'll respond to that and in most instances clear that infection, and no one ever knows it, because we don't know when animals become infected. What we know is when we can observe a large enough magnitude of the inflammatory response to detect it. And so that's kind of an interesting thing that will bring us to this microbiome research here in a minute, because a lot of the microbiome research has been done in absence of an understanding of the pathophysiology and immunological responses at the level of the mammary gland. Now, because the most common outcome of bacterial infection at the mammary gland is spontaneous bacteriological clearance. There's been numerous studies over the years that have gone into clinical cases of mastitis. Clinical cases means that the bacteriological infection has caused a large enough magnitude of inflammatory response that there's visible abnormalities in that gland. So swelling, abnormal milk. And people go in and they culture cases and they find a variety of pathogens. And so these are just, I don't know what, 8 or 9 studies from around the world showing what's cultured from milk samples collected from these mastitic glands. And the white bar down here is the one I want you to, the white segment, draw your attention to. Those are the culture negative milk samples from animals that have visible signs of inflammation, so clinical mastitis. And in North America, those typically run somewhere around 30 to 50%. So throughout my career, people have puzzled about, well, why would you get a negative, culture negative sample from an animal that has obvious mastitis? And have ascribed all sorts of reasons for that. Well, I'd like to say that what we've learned over the years is most culture negative cases are not the result of laboratory failure. Culture negative clinical cases, so again, clinical cases meaning you have abnormal, you have visibly detectable inflammation in either the milk or the gland, about 85% of these are truly culture negative. And what that is you've got inflammation, the abnormal milk, which is detected based on observation of inflammation after bacteriological clearance. And we've done a number of trials that shows inflammation is self-limiting. It lasts approximately 4.5 to 5.5 days with or without bacteriological clearance. So that's the immunology aspect of a bacterial infection. So these culture negative cases, clinical cases, in many instances are the result of spontaneous cure of opportunistic pathogens that don't have virulence factors that have allowed them to host adapt to the mammary gland. These have a great prognosis with or without treatment. We've done some negatively controlled clinical trials looking at this, and we find that all of the outcomes we looked at, quarter level recurrence, diminishment of the inflammatory response, and a return to a normal somatic cell count, and a rapid return to milk yield, much higher in culture negative cases. However, that's about 85% of them. They're not all truly culture negative. They're not all truly spontaneous cures. And I had a really interesting experience this summer, because this summer with my graduate student, Quinn Kolar, we induced strep uterus mastitis in 38 cows. We infected 38 cows. I think it might be the largest experimental induction with strep uterus done, as far as I can tell. And we have learned a lot doing this over the summer. So let me just show you some colony counts of strep uterus in cows we infected over time. So this is cow 10536. We infected this cow on day 1. The vertical axis is the number of strep uterus that we're recovering. We're collecting milk samples from these challenge quarters every day. And this is exactly what you want to see happen. So we infected her with about 10 to the 3 colony forming units on day 1. And you can see on day 2, you've got relatively low levels. These are logs, so 10 to the 2, 10 to the 3. And then by day 7, you're up to 10 to almost the 8th CFU. And this is what you expect to happen. And by the way, I put a little bar here because in these 38 cows that we've induced, typically when they get above 10 to the 5th is when they go clinical. The rest of the time they're subclinical. So that cow read the book, followed the directions, and behaved as expected. But we see these kind of cows. This is 91001. So we infected her. On day 2, she's at 10 to the 3.5, and on day 3, she's culture negative. Okay, so first we think, okay, spontaneous bacteriological clearance. But on day 4, here we are at 10 to the 6. Where are those bacteria, folks? Where are those bacteria? And then here's another cow, 5395. clearly infected her because the day after we infected her, she's got bacteria. Next 2 days, nothing. We're thinking, wow, this cow's got a great immune response. You know, she's cleared it. But here we are by day 6, we're at 10 to the 9th. So these are false negatives. Okay, there's a whole variety of reasons for that. And the microbiome, which I'm eventually going to get to here, is one of the things that's been hypothesized that may help and aid in some of these responses, right? So these are the culture negative clinical cases. Obvious inflammation, 85% truly culture negative, 15% false negatives. Now we have a different scenario with these chronic high somatic cell count cows. So these are culture negative cows from chronically inflamed mammary glands. So you've got evidence of chronic immunological stimulation because you maintain a high somatic cell count. And so you have a completely different scenario there when we look at these animals. Chronic high somatic cell count in a culture negative cow indicates failed inflammation and persistent intramammary infection regardless of culture results. And so these are often false negatives where the number of colony forming units that we're recovering in those milk samples is less than the detection limit in the lab. And that's what we were seeing in our strep uterus challenges. Apparently, I don't really know where those bacteria were, but there's some sort of indication likely of a robust inflammatory response, which keeps the number of pathogens below the laboratory detection limit. Now, here's where we get to the microbiome. People have been talking about these culture negative samples for years because microbiological culture of viable bacteria has been the gold standard for diagnosis of intramammary infection. And then we got this increasing proportion of culture negative samples. And at the same time that we're seeing this increased proportion of culture negative samples, we've got this explosion in the accessibility of these nucleic acid culture independent methods, which we can use to try to figure out what's going on in those culture negative samples. So we've seen an increasing interest in this milk microbiome. So what I'm going to spend the rest of my time here today discussing is really present an overview of the research to date on the bovine milk microbiome, although I will reference the human a bit, and to consolidate our current understanding of the relevance and the biological significance of this bovine milk microbiome. So I want to start with a few definitions. You'll hear me say microbiota and microbiome probably interchangeably just because I can't help myself. But the microbiota technically refers to the community of microbes, whereas the microbiome refers to the collective genomes of those microbiota, which a genome is, of course, the genetic material. It's really important for us to understand that that includes genomes of both viable and non-viable organisms. And the ability to identify a bacterial genome does not infer that a viable bacterial community exists. That is a very important issue as we look at the milk microbiome. We know very much so that these microbial communities of both culturable and non-culturable organisms have an extremely important function in many mucosal organs, especially organs such as the GI tract. We have some tremendously well-characterized human microbiomes, and those human microbiomes that have been very well characterized are especially those of the skin and mucosal organs. These systems have very well-characterized viable microbial communities. Let me repeat that. The microbiomes that have been shown to be very important in the health and homeostasis of human well-characterized communities have also well-characterized viable microbial communities. This is a very significant difference from the mammary gland. Okay, so that's an important thing for us to think about. The human microbiomes that in the GI tract and on skin are longitudinally stable. They're known to be influenced by some environmental factors. I upset mine a couple of weeks ago when I did too much wine tasting and suffered for a few days till homeostasis was reestablished in my gut. And they are known to interact with and influence localized immune responses. So we all hear a lot about these microbiomes. These microbiomes in these well-characterized communities can be, I shouldn't say can be, they are associated with both disease and health. And they've been shown to be very important for homeostasis in some systems. There's this really cool gut bacterium, which is found only in Japan, that can digest seaweed, which is of course part of that diet. Is that cool or what? There's gut bacteria, which can synthesize vitamin B2. And of course, you know, for hundreds of years, veterinarians and farmers have used transfaunation as a treatment for digestive disturbances of ruminant animals. So, you know, that's pretty cool. And then, of course, increasingly, we've got people working on feed efficiency to reduce methane emissions, and they're looking at manipulations of the gut microbiome in ruminants. Well, our subject today is, of course, the bovine milk microbiome. And there's at least 3 articles that have taken a look at the recent review articles that have looked at the bovine milk microbiota. And this is a figure from a JDS article in 2018. It's just a fantastic figure, very big, comprehensive review article. And in this review article, they talk about the colostrum microbiota. They've got a whole bunch of different OTUs that they discuss. They've got the milk microbiota, they got the teat canal microbiota, they got the teat apex microbiota, and then they got potential sources of the mammary microbiota. And one of the things that I just want to start with saying is we can find lots of genomes of bacteria in lots of places in the mammary gland. But in my opinion, it is not sufficient to simply characterize bacterial genomes from bovine milk samples and assume either functionality or dysbiosis. That is a huge overstep. Characterizing, and this is something that I think is really important for us as scientists to understand, characterizing the bovine milk microbiome is technically complex, and it requires a strong grounding and a strong multidisciplinary approach, which includes understanding of immunology, microbiology, molecular biology, and a practical understanding of issues influencing the diagnosis of bovine mastitis. When we isolate each of these areas, we make mistakes. And I think there have been a lot of mistakes made, and we've made some ourselves, as we have attempted to define a milk microbiota. And I'll show you where we're at today. This is my opinion right here, and I'll show you why as I move on. This is my opinion. Okay, so what do we know about the bovine milk microbiota? As a science, there's not much there, folks. There are relatively few peer-reviewed studies about the bovine milk microbiota. I redid this search, you know, I had to write the paper a few months ago, but I redid this search last week. This is a PubMed search, and I could find 36 studies across really time that expressively have looked at the bovine milk microbiota. And I want to say the early studies, many of those used producer collected samples, which are completely inadequate for this type of work. Okay, completely inadequate. So we don't even have 36 studies that I think you can compare. Another issue, and I'm going to get into this a little deeper, the methods used in these studies vary considerably. We've got huge issues with sampling, and we have virtually no replication. This is a huge problem in mastitis research in general, and is the issue of overinterpretation of unreplicated results. And I think this field is especially suffering from this. So let's get into how the microbiome is determined. We've got some very simple steps. We collect a sample, we extract DNA, we replicate amplicons, we sequence usually 16S, and then we do analysis. And I'm going to talk about some of the sources of error that have occurred and can occur as we go through this process. So let me start with the most important step of all. Aseptic collection of milk samples is absolutely vital to being able to understand everything downstream in the rest of the experiments. And so if we fail at this point, or if we cut corners at this point, nothing else matters because we're picking up contaminant DNA from environment in the skin. So I want to start with kind of the gorilla that is always in the room, that people are always debating, and it's at the first paragraph of every milk microbiome study is, milk used to be thought to be sterile, but it's really not. So I thought, geez, I wonder what the definition of sterility is. I actually never looked it up. So I looked up the definition of sterility, and the definition of sterility is the absence of viable bacteria. Well, almost all properly collected milk samples from non-inflamed glands have few viable bacteria that are recoverable, and that's almost regardless of method that you use. Okay, now if you leave them sitting around for a few days and then culture them, something will grow because it's a great nutrient source. But there's not many viable bacteria in properly collected milk. And I've personally cultured in my lab tens of thousands of samples using a variety of techniques. If you get them right, not much will grow. However, you can extract bacterial DNA from many of these sterile samples, but the issue we get into is the source and function of that microbial DNA is questionable. No one, to my knowledge, in those 36 papers we have of the milk microbiota, has yet demonstrated viability of these genomic elements, these nucleic acid-based sequences. And there are techniques to determine viability of bacterial DNA. They just haven't been done yet. And so at this point, in my opinion, we don't have evidence to conclude that milk from a healthy mammary gland is not sterile. In other words, doesn't contain viable bacteria. So I think this point isn't really too debatable yet. It may be in the future, but it's not yet. So we got many sample collection issues, which I think contributed to some of our confusion. Many microbiome papers state that they used NMC procedures to aseptically collect milk samples. And these procedures, these NMC procedures, which essentially mean that you used alcohol to clean off the end of the teat, they weren't designed for these techniques. They weren't designed. They were designed for culturing milk to look for initially Staph aureus and Streptococcus agalactiae. It is extremely easy to contaminate milk samples used for culture with skin and environmental bacteria. And even my graduate students whose careers depend on collecting clean milk samples, we get somewhere between 5 to maybe 15% contaminated samples even in their research animals. And it's up to 40% of cultured samples when you're having producer collected samples. This is a really cool study. It had nothing to do with milk microbiome, but it did have to do with PCR detection. So this is a study that was conducted in Finland and Holland more than 10 years ago when they were developing the pathoproof diagnostic test, which was now a widely used commercially available PCR test for pathogens in milk. They collected 1,000 porter milk samples using aseptic collection methods. And I don't have time to go into this full study today, but some of them were collected in Finland and some were collected in Holland. And the ones collected in Finland were way cleaner than the ones collected in Holland. And that's just buried in the paper. But I think that's pretty interesting. If you really want to do meticulous mastitis research, apparently choose Finland. Okay, so they had 780 cows in clinical cases, 220 milk samples collected from healthy cows. They cultured these in a lab using standard techniques. And here's the secret, folks. If you don't culture milk samples, but you get lab reports back, culture negative samples don't mean no bacteria were found. In microbiological labs for mastitis, non-significant growth means, oh, maybe there was a colony over here or a colony over there, but there's not enough that we think it's an intramammary infection. That's the reality. So a culture negative milk sample often contains some viable bacteria, just not enough of them. And so that's an important thing to think about as well. But they also performed this PCR test using PathoProof, which is a commercially available test. And I want to share what they found because I think this is informative as we look at the milk microbiome studies. So here is the number of bacterial DNA, so matched with primers, identifications in the culture negative samples. And okay, take a look at this graph. On the vertical axis is the percent of the samples. Here's the bacteria that were identified. They were in the primer mix for this test. And what I want to point out is 50% of the culture negative samples contained what at that time was being called coagulase negative staphylococci, non-Staph aureus staph. Today, this is called NAS, non-aureus staph. 50% of them contain non-aureus staph. About 30% contain C. bovis. And I just want to focus on those. Those are skin contaminants, right? They're also in the street canal. The street canal has a very microbiologically rich flora, definitely a microbiome present there. And they found a lot of this. And the thing I want to point out as well, in those samples, there were bacterial DNA that matched with many of the other primers. Two species were found in 23% of samples, greater than 2 species were found in 9% of the samples. So they're picking up a lot of bacterial DNA, regardless of having culture negative samples. But the types of organisms that they're finding are typical of those that if you swab skin, or if you put up and take a sample of the street canal keratin itself, you're going to recover these organisms. And then here's really a key paper that came out a few years later that we actually replicated in a microbiome study that I'll show a little bit later, where they actually went in and looked at the effect of these teat skin contaminants on recovery of non-aureus staphs. So this is a study where they used 2 different sampling techniques to collect milk samples. One was through the teat end, and one was right here, a puncture into the cisternal cavity. And this is likely my graduate student, mine and Laura Hernandez's graduate student, Stephanie Metzger, when we were replicating this on a microbiome study. So this is how you do this. You actually take a needle and you do a puncture into the cistern. You do need to tranquilize the cows if you want to survive on this one, by the way. So I want to show you what they did here. They compared 2 different cycling thresholds, and 2 different methods of collection. And what I'm showing you here is a proportion of samples that they recovered non-aureus staphs. So at a CT threshold of 34, 20% of the samples approximately contain non-aureus staph when they took them through the teat canal, but only about 8% contained it when they went through the used cisternal puncture. So, less than half. So they picked up a lot of non-aureus staph through that teat canal. When they went to 40 cycles in their PCR, they tripled the amount of non-aureus staph that they recovered with the traditional collection and they more than doubled with the cisternal puncture. And by the way, many milk microbiome studies require at least 40 cycles because we've got a low biomass sample. And so what we're looking here is replication of very few organisms. And if you ever look at what that 40 CT actually translates to, it's like, I tried to calculate this one time, it's like 1 trillion, okay, replicates. All right, so that sample collection has a big influence. Let's move into DNA extraction. So we get our milk sample, you've got to extract the DNA, but we got a low biomass sample, and that matters a lot. Determining the microbiota of low biomass samples is much more difficult than determining the microbiota from organs that have a rich flora. And there's many, many technical issues which you get into. And here's a study that says DNA extraction approaches substantially influence the assessment of the human breast milk microbiome. This is a study from Douglas in 2020. They had, in this particular study, they made both a mock microbial community, so they knew exactly how many bacteria and the types of bacteria, and then they used some regularly collected samples of human breast milk, and what they found was Significant differences in DNA yield and purity were observed between methods. The microbiota composition assessed by 16S rRNA gene amplicon sequencing also differed significantly with extraction methodology, including in the contribution of contaminant signal. And here's the take home, folks. Concerningly, many of the bacterial taxa identified here as contaminants have been reported as components of the breast milk microbiome in other studies. And I think the same caution can be applied to studies that we have done and others on the milk microbiome. And here's a paper. So I did my little search again as I prepared this. And here's a paper from September 2021. It's not in my paper yet that I've written to go along with this, but here's a paper called Bovine Milk Microbiota Comparison Among 3 Different DNA Extraction Protocols to Identify a Better Approach for Bacterial Analysis. This is a study done by the Cremonese group in Italy. And they compared 3 extraction methods using both a mock community, very similar to the human breast milk study, and 3 bulk milk samples. Bulk milk samples are full of bacteria, by the way. They're because, you know, they've come from multiple cows through the milking machine into the tank. I mean, you got a lot. And in comparing these different extraction techniques, they reported differences in DNA yield, abundance, and diversity metrics. I just put up here the graph of the mock community. You don't need to see these little numbers, but every color's a different OTU. Okay, so different genera or species. And so this is protocol 1, this is protocol 2, this is protocol 3. All you got to see is they got different results with different extraction procedures. And they concluded that it underlines the importance of the choice of a suitable protocol for DNA extraction, as the method can influence the 16S rRNA gene profiles. And so this is a significant issue for many of the milk microbiome studies. Virtually most of the studies use different extraction procedures, and that has an influence. So another issue we get into in this milk microbiome studies is milk just doesn't have a lot of bacteria in it when it's properly collected. And this is a study that I conducted with Laura Hernandez and our graduate student Stephanie Metzger, along with our collaborators Garrett Suin and others at the University of Wisconsin when I was still there. We collected milk samples using extreme aseptic technique. And this was so extreme. You just can't believe how extreme this was. Our graduate student Stephanie would put on 4 gloves to start, and she'd peel off the gloves on each one. The people in the milking parlor, we did this at the university, heard they hated us because it took like 15 minutes a cow to collect a sample. Okay, this was extreme aseptic procedures. And we did this at dry off, week 1 and 2 post calving, monthly to 150 days in milk. We identified the cows into different cohorts, et cetera. And because after months of trying to recover sufficient DNA amplicons, we ended up using 40 PCR cycles, which was what people were using when they were collecting them. And our overall sequencing success was 53%. Okay, that was our overall, and it was affected by the type of sample. When we took the samples from healthy cows, healthy quarters of healthy cows, okay, we had very low success, about 50%. When we took them from chronically infected cows, new infections, or animals that were positive, our success went up higher. So clearly, the presence of viable bacteria influences what you're going to recover from these samples. All right, just in case you're wondering if I'm going to be here forever, first of all, I have a little extra time because I didn't start on time. And second of all, I'm not doing the last 2 steps here. I didn't have enough time. So I want to talk a little bit about the PCR. And that's where I'm coming back to multiple PCR cycles amplify contaminant DNA. We saw that in our studies. In our initial studies, we're doing these 40 PCR cycles, and the #1 OTU we got was Halomonas. I'm like, Halomonas? I've been working in mastitis forever. I have never heard of Halomonas. So I look up Halomonas. It is a salt-loving bacteria. I'm thinking, salt-loving bacteria in the mammary gland, how's that working? And so then I'm like, this is weird. So I spend like 2 days like searching for Halomonas, and I find some paper from like the Estonian Journal of something like microbiology or something, and they say Halomonas as a contaminant intact polymerase. And I'm like, oh, okay. And yeah, Halomonas can be a contaminant in Taq polymerase. And right now, the ketome, how many of you heard about the ketome, right? The ketome is the trendiest thing ever. So here's a couple papers that how the ketome, in other words, contaminants in your reagents, influences the characterization of bacterial communities in samples with low bacterial biomass. Reagent and laboratory contamination can critically impact sequence-based microbiome analyses, and this is a huge problem in the milk microbiome studies. The reagent contamination has been hugely under-recognized issue in milk microbiome studies. Here's a paper from 2019. They took 278 milk samples from 9 cows. They did NMC collection. They used cows that had low somatic cell count, consistent bedding source, and they sequenced their no template controls. And 50% of their no template controls contained measurable DNA. And so here's their no template controls. This is the results of that study. Again, you don't have to see what all these OTUs are. Just look at this light yellowish color. This is their no template controls. These are their milk samples. Okay, you'll see the number 1 OTU they recovered was the same thing they found in their no template controls. Of course, you can subtract those if you sequence your no template controls and get some usable evidence. But many, many, many of the early studies, virtually none of the early studies on the milk microbiome have done that. So it's a significant issue in the research. So they concluded in this study, the Dahlberg study, evaluation of the composition and temporal stability of the microbiota in 288 milk samples was largely hampered by background contamination. And we observed an increasing impact of contamination with decreasing microbial biomass, especially when it's less than 10 to the 4th cells per ml. Very significant issue in low biomass studies. All right, just for a few minutes here to kind of get to the heart of the matter. What is the milk microbiome? How do we characterize it? Well, first of all, I just want to remind everybody that I'm not getting into the downstream processing. I just don't have enough time for it. But different sequencing platforms have differing abilities to identify organisms at the genus, family, order, class, phylum, or kingdom. So you may have a phylum, you may have a class, you may have an order, you may have a family, you may have a genus. In some instances, more recently, you get some species. So really difficult to compare because you get different levels of discrimination. I also want to comment on this, the way we characterize the milk microbiome. I've looked a little bit into this. How did we get to these diversity enrichment and richness stats? Like, because I wasn't familiar with those before I started working in milk microbiome a little bit. And this is really interesting history because the community composition of richness and diversity is based on the assumption that healthier communities are more diverse and more abundant. They have more species, they have greater number of OTUs, they have a greater evenness. And this actually comes out of how ecologists characterize like wildlife and trees and all of this, which I find really interesting. It's important to recognize that these statistics are calculated on a relative basis. So if you get a new intramammary infection with a lot of growth of bacteria, you know, your relative proportion of the others will decrease. So you're going to have reduced diversity. That's going to happen. That's how you calculate them. And another thing that we really need to think about is the assumption of the value of increased diversity and richness has not been validated for low biomass organs. We just don't know if that applies. And I think that's important as scientists, not just to make that leap, but somebody needs to go in and figure out if that applies. So what do we know about the biological relevance of bacterial DNA extracted from milk? I'll bring you back to this potential sources of mammary microbiota from the Derek Shani paper in 2018. They show here's some potential sources: bedding, milking equipment, hands, flies, cross-sucking, lots of different sources. But the thing that nobody has demonstrated is, are these genomic elements, the microbiome, are they functional, or are they remnants of a successful immune response? Are they contaminants? Are they bacterial DNA trapped within keratin? Are they bacterial DNA within leukocytes? Remember, I infected animals and couldn't find them a couple days afterwards. Where were they? They had to be in leukocytes somewhere or way below my level of detection. Are they viable? And is there really the thing that's really been out there a lot is there a core mammary gland microbiota? And this concept of the functional core mammary bovine milk microbiome is very controversial with immensely varying viewpoints. Here's two different papers: Mammary Microbiota of Dairy Ruminants, Fact or Fiction by Pascal Renard's group. This is really, anybody doing science should read this paper, I think. It's an outstanding paper. And then here's a fairly recent paper, which completely different viewpoint. A core microbiota dominates a rich microbial diversity in the bovine udder and may indicate the presence of dysbiosis. So here's the conclusion from the Renard paper. This article develops the logical implications of a paradigm shift, the shift to dysbiosis, and shows how this concept is incompatible with current knowledge concerning innate and adaptive immune system of the bovine mammary gland. And then here's the contrasting viewpoint. The overall composition of the udder microbiota was highly diverse and significantly different between cows and between quarter. And furthermore, we introduced a novel definition of a dysbiotic quarter at the individual cow level, completely opposing viewpoints. Well, what do we really know? This is from a table in Derek Shawnee's review article, and I summarized it by just making a vertical axis from not identified OTUs up to most abundant. And you've got the teat skin apex, the teat canal, and milk. And what you'll notice is wherever you've got milk, what you've got is a whole bunch of bacteria that also live in the teat skin and the teat canal. And when you don't have those, you don't have them in milk. And so I think we've got to just step back a little bit and think more about doing some fundamental research. The origin of these bacterial genomes found in milk is not well defined. Most of the common genera colonize teat skin. They're found in farm environment. And in work that we did while I was at the University of Wisconsin, we demonstrated that exposure to bedding strongly influences the recovery of bacterial genomes in bovine milk. This is a study, again, Stephanie Metzger as part of her PhD dissertation. We went to one farm, a University of Wisconsin farm, and this is actually a study where I collected samples. I actually went to the farm and collected samples. It was like me and Stephanie in an undergrad. And so I feel pretty confident in this because I was there on this one. And so in this, we had 4 groups of animals in the same barn on 4 different bedding types. And I just remembered we had this long-term experiment somebody else had going on. And I said, hey, can I have 20 of those cows? And we went up there and we collected extremely aseptic samples. We took cisternal punctures and we took those samples back and sequenced them. And this is the result. Here's the clustering on the PCA. So you can see that the 5 samples, quarter samples from the cows on sawdust, the cows on manure solids, the cows on recycled sand, and the cows on new sand are all clustered. And the cool thing here is that I think gives a lot of real validity to this study is this is recycled and this is new sand, and those 2 clusters are together. So whatever's in the mammary gland is highly influenced by what the cows are standing on. And these were really healthy cows collected using extreme aseptic, and this is the cisternal puncture samples. All right, I'm going to finish up here in a couple minutes just by talking about the concept of dysbiosis. So this concept of dysbiosis is kind of what everybody's trying to find. If we have a dysbiosis, some unbalance in the composition of the microbial community, then it affects the well-being and health of that organ system. And this concept of dysbiosis is extremely well defined for organ systems that have robust, abundant, functional commensal flora. And so like the gut, the skin, whatever, if we disrupt that, we know that these dysbiotic communities are associated with several disorders, IBD, obesity, and diabetes in humans. I would argue that the theory of dysbiosis in the mammary gland is highly speculative. And the composition of the microbiota that we can recover, either viable or non-viable, may change after intramammary infection, but that could become the pathogen becomes the dominant species, or as Suvi's group has argued in Finland, The milk composition changes when you have intermammary infection, which may favor growth of other bacteria. And so it's a highly complex system to assess. The Tappan Group conclusion on this article in 2019 said it has been suggested that intermammary infection would be a consequence of a dysbiosis of the mammary gland microbiome. And she concludes that with saying no direct scientific evidence for the dysbiosis theory has been promulgated. So what do we know? Bacterial DNA can be recovered from some properly collected milk samples, but DNA extraction is often not successful and it requires many, many PCR cycles, which is indicative of very low density of microbial DNA in samples. The sources of that bacterial DNA, at least some of them, are likely environmental. Only samples collected using extreme aseptic techniques or cisternal puncture really are suitable for evaluation of the milk microbiome. Very, very important. They need to be quarter samples. There needs to be extreme care taken in collection of these samples. There are numerous differences in methods that preclude comparison of results among studies, and many of the early studies we simply can't use because of some of these technical difficulties. The influence of farm environment, sampling technique, lab methods, and other factors need to be considered. And I believe it is much too early for us to draw conclusions about the significance of a commensal mammary gland microbiota. That said, a commensal teat canal microbiota clearly exists. So for my final conclusion, bacterial DNA is found in milk samples. Some is viable, but viability for most is questionable. We have decades of standardizing methods for milk microbiological culturing that have given us excellent ability to understand the results of those milk cultures. But we don't understand the impact of using a variety of non-standardized lab techniques on results of these molecular methods. I think the technical issues related to these low biomass samples and a lack of standardized techniques have impeded our progress. And you know, one of the things that's a huge difficulty in the field we work in is a lack of replication. We really need some replicated studies from multidisciplinary groups who understand the entire gauntlet, really, or process from infection all the way through kind of health or illness. So with that, I want to really thank my collaborators, students, and others at both my former institution, the University of Wisconsin-Madison, that includes Laura Hernandez, Garrett Sewin, Terry Walker, Joseph Scarlupa, and Stephanie Metzger, and my current collaborators at Michigan State, including Shannon Manning, Ron Erskine, Quinn Kolar, Giuliano Goncalves, Li Zhang, and in memoriam, Lorraine Sardillo, who passed away last month. Thank you.